Hello, everyone. Today I'm going to give a quick overview of Chapter 11 titled Genome Alterations, Mutations, and Epigenetics. Today I will be covering several sections from Chapter 11, so not all sections will be covered. We're going to overview Section 11.2, which talks about mutations, 11.4, which further dives into the types of mutations. Some can be spontaneous, others induced. 11.5 will cover mutations at the molecular level, so what is going on at the level of the DNA. 11.6 covers mutations and how they can be repaired, so there are repair mechanisms for mutations that occur during any moment during the cell cycle, and also how DNA can be modified by epigenetic changes, meaning that the expression of a gene can be changed not just by mutations, but by this other mechanism called epigenetics, which we will cover at the end of this section. So let's talk about mutations. Mutations are changes in DNA by the nucleotide sequence. They can be a single nucleotide change, a depletion or insertion of one or more base pairs, or an alteration in the structure of a chromosome. And so mutations in body cells, for example, just somatic cells, do happen often, and they can be passed on to other cells during cell division, but this is not the type of mutations that get transferred to future generations. And then there are another set of mutations which occur at the germ cells, meaning that they are the sex cells. And these type of mutations are the ones that continue being passed on to future generations. Some mutations can be spontaneous, others can be induced. So, for example, a spontaneous mutation are errors in normal cellular processes such as DNA replication. So, there can be an error during DNA replication that can cause a mutation. Some can be natural changes in the molecular structure of the bases of the DNA, so a nucleotide can be exchanged. Other types of mutations can be induced, meaning that chemicals, maybe from the environment or some type of chemical agent that was absorbed by a cell, can attack the DNA and disrupt it. And so some examples include radiation and chemicals. So, for example, a spontaneous mutation can occur at the level of DNA polymerase. So, if you remember, DNA polymerase is involved in DNA synthesis. It is the main enzyme that fuses the nucleotides together. Every now and then, DNA polymerase can accidentally pair the wrong nucleotide. So, here's a picture showing that A is being ready to be paired, and we know that it should be paired with the nucleotide T. However, in this case, it is accidentally paired with C. And this mutation, for whatever reason, seemed to work, even though they are not the right base pairs, and DNA polymerase continues. And so this is rare that this occurs, just because, and we'll look at this later on in the PowerPoint, but because DNA polymerase itself is designed to proofread, but yet this type of mutation can happen, and so no system is perfect. So one possibility for nucleotide base pairing when it comes to base pairing of the wrong nucleotides is something that is called tautomeric shift. And basically this is one of the molecules, for example here, guanine is basically its conformation is reversed. And because its conformation is reversed, these hydrogen bonds that are formed between thymine and guanine are possible, and this can cause a forced base pairing. DNA mutations can also be caused by radiation, and radiation is all around us, and it's basically energy traveling through space. And so even our bodies radiate energy, the food we eat, the bricks in our houses, that all has background radiation. And there's also radiation that is created by human activity. For example, like apparatuses for medical testing, nuclear power, consumer goods. There's all sorts of forms of radiation. And basically, radiation is energy being transferred to DNA, and that DNA absorbs the energy and causes some type of damage. So it can be base pair damage, a single strand break, or a double strand break. And so it just depends on how much energy was absorbed when it was exposed. So here are some examples of radiation. So there's cosmic, terrestrial, so here on Earth, this is from space, internal, medical x-rays, nuclear medicine, consumer products, and radon is the biggest source of radiation. Other induced agents that can cause DNA mutations are chemicals, and there are about 500,000 different chemical compounds that are used for manufacturing and packaging, food, building materials, and so forth. And a lot of these compounds have been linked to DNA damage. And so here are some examples at the bottom: carcinogens, like in cigarettes, processed foods and preservatives, chemicals that are found in cleaning products and cosmetics as well. And so these kind of chemical mutagens can cause different types of DNA damage. And so it can be a DNA strand break or a base pair break. And so it just depends on how the mutagen works. So there has been an issue lately about irradiated food. And so, since we know that radiation is damaging to DNA, that also means that it can kill microbes because microbes contain DNA as well. And so, radiation has been used to basically preserve food and kill contaminating microorganisms. And so radiation basically is like an alternative to using chemical preservatives, which are also used to basically kill off microbes that are unwanted in food. And so, although radiation has been okay for the past few decades and there hasn't been any issues with it, there is growing concerns as to how radiated food can affect us. And some people point out that treatment with radiation may create microbes that are resistant to that type of treatment. And so that's a good point to bring up. And so I don't know what all of you think about that, but what do you think about having food that's irradiated and then you eat that? Do you think that's a problem? All right, let's move on to section 5, which is mutations at the molecular level. And so now let's look at what goes on at the level of DNA. And so we've discussed earlier that mutations can involve substitutions. And so this is a nucleotide substitution, which therefore, so if you have a nucleotide substitution, like what it shows in this picture here, so A was swapped for C, that can result in a whole different amino acid being produced. And so that can potentially, change the structure of a protein depending on whether it's on the sweet spot of the protein. And so mutations can be substitutions, insertions, deletions of one or more nucleotides in a DNA molecule. All right, so let's go over a type of mutation called frameshift mutation. This is probably one of the most damaging types of mutations. And so in this case, a frameshift means that either the insertion of an extra base or the deletion of one or more base completely changes the frame. And so from then on, after that frame, a completely different amino acid can result. So for example, here's a normal strand of DNA. And in the bottom, if you notice here, an extra nucleotide was added. So this would have normally been tyrosine, except a whole new nucleotide was added, which is uracil. And because we have an extra, from now on, there's a frame shift. So now this is one codon, this is one triplet, right? We have 1, 2, 3. And now this is another triplet, 1, 2, 3. And we have another triplet, 1, 2, 3. And this extra one right here, in the end, will continue, continue on. But the point is that this extra nucleotide here completely shifted where the codons start and stop. And so as a result, you end up with different amino acids down the line. So if that extra nucleotide was not added, you would have tyrosine, tryptophan, and asparagine. But since this extra nucleotide was added and it shifted the codons, and so now we go from here to here to here. Now you end up with different amino acids. So you have leucine, leucine, glutamic acid, and so on. And so that can completely change the type of protein that is resulted from this. And so that's very damaging to the protein. A missense mutation is just a single nucleotide change. And so it doesn't frame shift. It doesn't change how many nucleotides are there in total. It's just like a swap. So in this case, this is if you remember from sickle cell anemia. In this case, a single swap from T to A. So here, T to A, change the amino acid from glutamate to valine. And here's another single swap. So it changed the amino acid to lysine, and another single swap, which changed the amino acid to alanine. So in this case, the phenotype may be defective, or it may not cause any type of defectiveness. So all just depends on where on the protein is that mutation. If it's on the sweet spot, and let's say that sweet spot, what I mean by sweet spot is like a certain area of the gene that is very important for its three-dimensional structure. So if you hit one of the three-dimensional structure areas, you may completely morph the protein. You may completely change the DNA structure just by this one point mutation. But if it's not on a sweet spot, if it's on a area in which it doesn't affect the protein, the way it's folded, or if it doesn't affect like its catalytic region, then it may not cause any type of disorder or anything detrimental whatsoever. And so a missense mutation is less detrimental than a frameshift mutation. And there are also something that's called a sense mutation and a nonsense mutation. Let's talk about nonsense mutation first, since I have it here on this picture. A nonsense mutation is basically changing the code, a triplet code. It's changing it from one that would have coded for an amino acid to one that encodes for a stop codon, meaning that instead of the protein being translated further, it completely stops prematurely. And so if you have an mRNA that stops prematurely, you know that damages the protein. Either the full thing isn't translated, or it doesn't, well, basically the full thing isn't translated in this case. The sense mutation, the one that's above, is basically changing the stop codon to one of an amino acid. And so in this case, a sense mutation, you would have the mRNA would be like a run-on. So instead of stopping where you're supposed to at the end of the protein, it would keep translating the rest of the amino acid code that really shouldn't be there. Why? Because simply the stop codon was changed from a stop into an amino acid. So this is a run-on. So a sense mutation is basically like a run-on of a sequence being translated, whereas a nonsense mutation is a premature stop. So as the mRNA is being translated, it prematurely stops, basically chopping off half of the protein, or just depending on where it lands, right? Now we know that DNA is extremely vulnerable to becoming damaged. We learned that the environment can cause damage to DNA, as well as the natural processes that may have gone wrong during, let's say, DNA synthesis can also cause damage. And so, knowing this, we know that basically DNA is very vulnerable to becoming damaged. And so, there must be some repair mechanisms that are in place to basically repair DNA in case this happens. And indeed, such mechanisms do exist, and we'll cover a few in this section. But first, let's talk about what happens to cells when mutations accumulate and there is no repair. So here is three possible fates. One is that cells can become dormant, and so they just are no longer active anymore, and so this condition is called senescence. So they're just getting older, they become dormant. Another strategy for getting rid of all of this DNA damage accumulation is for cells to commit self-suicide, which is funny to say that, but they actually do self-destruct, and this is called apoptosis. And so, for example, we know that sun, if you're exposed to too much sun, uh, it gives off a lot of radiation, which can be damaging to your skin. And so, if you've ever sunburned and if you notice your skin starts peeling, that's basically the cells that have been damaged that are basically dying and coming off of your first layer. Another possible fate of having too much DNA damage is something that is called cancer, which a lot of us are familiar with. We've heard about it, or maybe we know somebody who's gone through cancer. And so, in this case, this is basically cells escaping the normal controls of the cell cycle. And so, so much damage has been done that these cells no longer respond to their environment. They kind of just do their own thing. And so, that is called cancer. And so, if DNA repair mechanisms weren't constantly in place, cells can be subject to damage very easily. There's quite a bit of DNA damage going on even per hour. And so here's a table of the types of DNA damage that we're subject to, as well as the rates or the events per hour. And there's also DNA repair rates, meaning that there's a lot of DNA repair mechanisms going on within our cells also per hour. And so, as you notice, there's a lot of repair mechanisms going on constantly. And so that just means DNA is very vulnerable to becoming damaged. And so let's talk about a DNA repair mechanism that involves DNA polymerase. And so we know that DNA polymerase is involved in DNA synthesis. And so it's involved in fusing nucleotides together. And luckily, this type of enzyme is also great at proofreading. And so it's basically reading 20 bases per second. And if an incorrect nucleotide is added to the new strand, the enzyme can recognize that. And it can also basically swap out the incorrect nucleotide. So let's say that the incorrect nucleotide here is C. It can swap it out for the correct one and basically correct the mistake. And so it's just great as a proofreader. We also know of another type of repair mechanism in which several enzymes are involved in detecting just basically the slightest distortion in DNA. And so we know that DNA is like a tightly wound double helix, and any slight distortion in the helix. So, for example, if it's a little bit, the curl is a little bit longer or a little bit shorter, there are enzymes that can detect that distortion and basically snip off the part of the DNA that is causing that distortion and basically recruit DNA polymerase to repair the nucleotides that should be there to put it back into its original shape, which I think is really neat that our cells have the ability to do this. There are diseases associated with damaged DNA repair mechanisms. For example, the genetic disorder xeroderma pigmentosum is caused by mutations in the DNA repair genes themselves. And so, if an individual is born with a mutation in the DNA repair genes, then that can be very detrimental. So, these individuals that have xeroderma pigmentosum cannot be exposed to the sunlight because the sun has very high energy and can basically destroy the DNA. And so we talked about that earlier, how radiation can affect DNA. And so these people are extremely sensitive to sunlight, and they often have dry, flaky skin and pigmented spots that can develop into skin cancer. And so this is an individual affected by xeroderma pigmentosum, and as you can see, she has a lot of spots on her skin. So all of the previous types of mutations that we talked about all result in a different type of gene expression. However, there's another way to manipulate gene expression without it being done by mutations. And this is something that is called epigenetics or epigenetic changes. And so epigenetics are basically the changes in gene expression without affecting any part of the DNA sequence. And so this is not damage done to the DNA. This is just a change in the way that genes are expressed. And so a gene can be expressed more than normal, or it can be expressed less than normal, or not at all. So, for example, there are some people that develop lactose intolerance later on in their lives. That can be an epigenetic change, and so the enzyme that normally breaks down, let's say, lactose, is no longer being produced at the same levels or being produced at all. And again, that can be through an epigenetic change. And so epigenetic changes are reversible chemical modifications of chromosomal DNA, such as methylation of bases, which we have talked about in other chapters, or histone protein modification. So if we remember from previous chapters, DNA is basically tightly coiled around these proteins called histones. And the more tightly coiled they are, the harder it is for a gene to be expressed because the promoter region is hidden. However, if if there's a modification done to the histone, so for example, adding a methyl group, an acetyl group, or a phosphate group onto a histone can distort the histone shape, and as a result, uncoil the part of the DNA that should be expressed. And just as histone modification itself is a type of epigenetics, and so you can control gene expression just by modifying the histone. And again, that's an example of epigenetics. And so the DNA in this case is not being altered, the nucleotide sequence is not being altered. It's more of the way that the gene is being expressed. And so another form that we had talked about before is DNA methylation. And so DNA methylation are basically methyl groups that are added onto DNA. And those methyl groups themselves can either block the promoter site or promote the binding of DNA polymerase or other enzymes that can promote the binding for gene expression. And so in this case, DNA methylation can completely silence the gene being expressed, or it can enhance it, but it definitely changes the way that it's being expressed. And so this again is a type of manipulation of gene expression that is not done by a mutation. And so that is the end of the review for this chapter. There are still more stuff in there that you should read about that I didn't completely cover, like that I'm asking you for your that I'm asking you to do for your study guide. However, you're only responsible for all of the information on the study guide. So if you decide to read the other sections in the chapter, which I think are very interesting, by all means, go ahead and you can learn from it. But I just won't be testing you on everything. The only thing I'll be testing you on is what I cover in the study guide. And so I created basically this little mini lecture that I'm giving you right now just to guide you through the chapters. But remember, I also provide you with other videos and other ways for you to be able to better understand the information. If you need more help, please come by to office hours, which is on Thursdays from 11:10 a.m. to 12 p.m., so don't hesitate to come by. You can also send me an email. The best way lately to get a hold of me is actually through the email from Canvas, straight on the inbox. And also there's a section, I think it's called Pronto, in which you can send me a chat. And so I tend to respond to those the quickest. But sending it to my personal email, the sdccd.edu. Lately, our email system has been having a lot of problems, and it's been down. So I just suggest to stay away from that email. If you need to get a hold of me, do it through the Canvas inbox, send me an email through there, or do it through Pronto.